Welcome to our second film on photosynthesis, where we will be looking at the requirements for photosynthesis. In the film after this, we will be looking at how some of these factors can affect the rate of photosynthesis. That's right, we've already looked at how synthesizing glucose from carbon dioxide and water using sunlight is vital for all life on Earth, not just because of the sugars that are produced, but also because of the oxygen that is produced as a byproduct. The following components are required for photosynthesis: light, carbon dioxide, water, heat, minerals, and chlorophyll. Photosynthesis takes place in the daylight. The sugars that are produced are used for cellular respiration constantly, day and night. Obviously, whilst it is daylight, extra glucose is being produced, and this is stored as starch until it is required. In order to confirm that photosynthesis had taken place, we need to test for the presence of starch. That's quite simple. So first of all, let's look how you test for starch. Take your sample leaf and boil it in water for 2 minutes. This will kill off the leaf and stop any more metabolic processes. It will also rupture the cell walls. Now place the leaf in a test tube of ethanol, which is heated in a water bath. The chlorophyll can now be absorbed by the ethanol. After a few minutes, remove the leaf and rinse it. This will remove the chlorophyll and make the leaf less brittle. Iodine solution is orangey brown, but when it reacts with starch, it turns blue-black. This is a positive test for starch. If starch is present, it is because glucose has been made, and this is what confirms that photosynthesis has taken place. We will now highlight how this can be used for each factor being tested. We will start by looking at light. Take a healthy living plant and place a piece of black paper on a leaf using a paper clip. Be careful not to damage the leaf. Now leave the plant in the sunlight and return to it after 2 to 3 days. Before you do anything else, make a rough diagram of your leaf to show which part was placed under the black paper, and then carry out your test for starch. This time you will see that the part of the leaf that was covered in black paper is now orangey brown, and the other part of the leaf is now blue-black. But what does this show? The covered part of the leaf did not receive any sunlight. Those cells could not produce new glucose, but they carried on respiring. The starch in these cells was converted back into glucose and used in respiration. The other part of the leaf that turned blue-black tested positive for starch, indicating that glucose had been produced and confirming that photosynthesis had taken place. The only difference between the two parts of the leaf was the presence of sunlight. From this experiment, we can prove that sunlight is required for photosynthesis. Now let's see how we test for the relevance of chlorophyll. You are going to need a variegated leaf, one that contains chlorophyll and non-chlorophyll sections. No need to wait for anything here, we can just dive straight in and do the starch test. But don't forget to make a diagram of your leaf first, so you remember which bits are green. The positive test shows that chlorophyll is required for photosynthesis. Now let's look how we test for the importance of carbon dioxide in photosynthesis. Take a look at this plant. We have bagged up 2 leaves. One of the bags contains soda lime in solution. This will absorb all the carbon dioxide. The other bag contains hydrogen carbonate. This releases CO2 into the atmosphere contained in the bag. Once again, we will leave the plant in sunlight for a few days to see what happens. After 2 days, carry out the test for starch. Once again, a blue-black leaf will confirm the presence of starch, and this time we will see that the test is positive for the leaf that had a rich supply of carbon dioxide, and not for the leaf that was starved of carbon dioxide. Water is a limiting factor for photosynthesis, but we cannot test it in the same way. Water is not just vital for photosynthesis, it is vital to maintaining a healthy plant life and structure. Plants wilt when there is not enough water. If we completely remove all water, the plant will die. This will not present an accurate test. But it is worth knowing that if there is a reduced water supply to a plant, the stomata will close up, reducing the availability of carbon dioxide, so photosynthesis was stopped temporarily. at least until the water supply improved. We are down to our last 2 tests, and we are going to do something a bit different. The need for minerals can be tested by growing some seedlings in different mineral concentrations. We are specifically looking at nitrates and magnesium. Both of these are required to make chlorophyll, so their availability does not directly affect photosynthesis, but they can limit the availability of chlorophyll, and as we have already seen, this is vital for photosynthesis. This image shows that with magnesium and nitrates, the plant will be healthy and grow to its full potential. It's worth bearing in mind that nitrogen isn't just used for making chlorophyll. It is also used for making amino acids, enzymes, and proteins. Heat is more complex. There are varying degrees of heat. In our next film, we will look at the relationship between heat and the rate of photosynthesis. We will also look at how the rate of photosynthesis is affected by other factors. Now